Protein synthesis. Translation. What is translation? Translation is the process by which a protein is synthesized from the information contained in a molecule of a messenger RNA. During translation, an mRNA sequence is read using the genetic code, which is sets of rules that define how an mRNA sequence is to be translated into the 20-letter code of amino acids, which are the building blocks of proteins. The genetic code is a set of 3-letter combination of nucleotides called codons, each of which corresponds with a specific amino acid or stop signal. Translations occurs in a structure called the ribosome, which is factory for the synthesis of proteins. Translation is the RNA directed synthesis of a polypeptide. Translations involves mRNA, ribosomes, ribosomal RNA, transfer RNA, genetic coding codons. The genetic code. Genetic information is encoded as a sequence of base triplets or codons DNA molecule. Codons 3 base code for the production of a specific amino acid sequence of the 3 of the 4 different nucleotides. Since there are 4 bases and 3 positions in which codons, there are 4 times 4 times 4 equal to 64 possible codons. 64 codons but only 20 amino acids, therefore must have more than 1 codon. 3 of the 64 codons are used as stop signals. They are found at the end of every gene and marked on the end of the protein. 1 codon is used as a start signal. It is the start of every protein. Universal in all living organisms. A codon is a messenger RNA, is either translated into an amino acids or serves as a translation start or stop signal. Transfer RNA consists of a single RNA strand that is only about 80 nucleotides long. Each carries a specific amino acids on one end and has an anticodon on the other end. As a special group of enzymes pairs up the proper tRNA molecules with their corresponding amino acids, tRNA brings the amino acids to the ribosome's 2-dimensional structure. 3-dimensional tRNA molecule is roughly L-shaped. Ribosomes. Ribosomes facilitate the specific coupling of tRNA anticodons with mRNA codons during protein synthesis. The 2 ribosomal subunits are constructed of protons and RNA molecules named ribosomal RNA or rRNA. Building of polypeptide structure. Building a polypeptide. We can divide translation into 3 stages: initiation, elongation, and termination. The EUG start codons is recognized by methynyl tRNA or MET. Once the start codon has been identified, the ribosome incorporates amino acid into polypeptide chain. RNA is decoded by tRNA molecules, which each transport specific amino acid to the growing chain. Translation ends when a stop codon EUAA, UAG, EUU is reached. Initiation of translation. The initiation stage of translation bring together mRNA, tRNA bearing the first amino acid of the polypeptide and the 2 subunits of ribosomes. Elongation of polypeptide chain. In the elongation stage, amino acids are added by 1 to the preceding amino acid. Termination of translation. The final stage is the termination when the ribosomes reach a stop codon in the mRNA. Translation. The final steps in translation is termination. When the ribosome reaches a stop codon, there is no corresponding transfer RNA. Instead, a small protein called a release factor attached to the stop codon. The release factor causes the whole complex to fall apart. Messenger RNA, the two ribosome subunits, the newly polypeptide. The messenger RNA can be translated many times to produce many protein copies. Structure of translation. Post-translation, the newly polypeptide is now floating loose in the cytoplasm if translated by a free ribosome. It might also inserted into a membrane if translated by ribosome bound to the endoplasmic reticulum. Polypeptide fold spontaneously into their active configuration, and they spontaneously join the other polypeptide to form the final proteins. Sometimes other molecules are also attached to the polypeptide, sugar, lipids, phosphates, and etc. All of these have a specific purpose for protein functions.